Okay, hi everybody. Welcome to Living Resources Shellfish session. I'm Sarah Babulas, Habitat Project Coordinator for the Partnership for the Delaware Estuary. Thanks for coming. If you have any general questions during the session or tech issues or anything like that, please feel free to reach out via the chat and our tech help people will help answer your questions, tech related or otherwise, so please reach out. Just wanting you to know that all our sessions are being recorded and that chat threads will be saved. The recordings will be available on Sked and YouTube and our website in the next couple weeks. There are two opportunities to ask questions to our presenters today. Each speaker is given 15 minutes, and if they don't use their whole 15 minutes, at the end, we will open it up for questions via the Q&A box. So please enter your questions into the Q&A box. At the end of the whole session, we have 15 minutes that we'll open it up to for all our presenters. Actually, I have 10 minutes because we have a lightning talk in this session. And you can do those also via the Q&A box, or we encourage you to use the raise hand feature and we can call on you so we can hear your voice. Kurt Chang from PDE is our Q&A and he will be handling the questions during this session. Okay. So now I'm going to throw it over to Des Khan, who is our moderator for the session, to introduce himself and our first speaker. Go ahead, Des.
Okay. Hello, I'm Desmond Khan, and I'm going to be moderating the session today. Our first speaker is Archi Howlader from the University of Maryland Center for Environmental Science, and the title of her talk is Prediction of the Salinity History of Oysters in Delaware Bay. Go ahead, Archie. I think you need to unmute yourself, possibly.
Yeah. Thanks. So hi everyone. Today I'm going to talk about predicting the scientific history of oysters in Delaware Bay. So my name is Archi Howlader. I am from University of Maryland Center for Environmental Science. I'm currently doing my master's, and my supervisor is Dr. Elizabeth North. So let me.
Looks good.
Today I'm going to talk about first about the SIGO program, which I am funded by, and then I'm going to talk about research objectives and data sets, and then I'm going to talk about regression model, and then I'm going to talk about model testing, and then about salinity estimates for SIGO, and then I'm going to discuss about our next steps. So what is SIGO? SIGO is Selection Along Estuary Gradients in Oysters. It is an NSF-funded project. It has 3 PIs. One is Dr. Matthew Harry, who is from Cornell University. Another one is Dr. Daphne Mundo, who is from Rutgers University. And another one is Dr. Elizabeth North, who is from UMCS. So the overall goal of this project is to investigate the phenotypic and genomic consequences of selection in oysters. So the main hypothesis behind it is that low salinity stress can structure the population genetics of eastern oysters during the larval or early post-settlement stages. So now I'm going to talk about my research objective in this project is to predict the daily salinity history of oysters in Delaware Bay and develop the model with accuracy within ±2 PSU. So our basic approach is to access and curate historical data, conduct time series analysis, and then develop models to predict salinity over oyster beds, and then collect field data to calibrate and validate model. So now I'm going to talk about historical datasets and station location. So we have narrowed it down to 3 main datasets. One is Delaware River at Reedy Island Jetty, Newcastle County dataset, which is from USGS. I'm going to refer it as Reedy dataset throughout my talk. And I have 5 oyster bed stations. One is Hope Creek, another one is Arnold's, another one is Cohency and Sherrock and New Beds. I have 2 datasets available which took measurements in this 5 oyster bed location. One is US Army Corps of Engineer and Versa dataset. I'm going to refer it as ACOE dataset. And another one is Huskin Shellfish Lab Oyster Bed Monitoring Program dataset. I'm going to refer it as Huskin dataset. Here is a comparison of Armicop dataset, Reedy dataset, and Haskin dataset. In the x-axis, we have time. In the y-axis, we have salinity. And the first panel is for year 2012, and second is 2013, and then 2014, then 2015, and 2018. To save time, I'm going to focus on 2012. So here in the x-axis, we have time. In the y-axis, we have salinity. And the blue lines are representing Reedy salinity. So the first column is for Hope Creek. So the orange colors are representing Army Corps measurements of salinity in Hope Creek Station. And the black dots are for Haskin Lab measurements at Hope Creek Station. And then second one is for Arnold. The green lines are representing Army Corps measurements at Arnold Station, and the black dots are representing Haskin Lab measurements at Arnold Station. And the red lines are representing Army Corps measurement at Cohency. Pink lines are representing Shell Rock, and the yellow lines are representing New Bed. And the black dots are for Haskin measurements at that stations. So here we can see that Haskin Lab measurements usually fall into the Army Corps measurements at each respective oyster stations, and usually the Reedy oyster bed station salinity measurements usually follow a similar pattern as Reedy station. So the overall goal is to create a regression model that predicts salinity over each oyster bed using salinity at Reedy point. So now I'm going to talk about the regression model. So the basic summary of the steps are to convert time to GMT, and then shifting tidal basis and matching time series, and then fitting multiple nonlinear regressions to predict salinity over oyster bed given the salinity at Reedy point, and then select the best fitting model. So the best fitting model was logarithmic in shape. So here is the model for Hope Creek. In the x-axis, we have Reedy salinity estimate. In the y-axis, we have Hope Creek salinity measurements, which were done by Army Corps. So the model is salinity at Hope Creek is equal to A plus B into log in the bracket salinity at Reedy minus C. So what is ABC? For A, for increasing or decreasing A, the graph will go up and down. For increasing or decreasing C, the graph will go left or right. And for B, the graph will be steeper. So for Hope Creek, the 95% prediction interval width was 4.4. For Arnold's, we have the similar kind of model, and the 95% prediction interval width is 4.6. For Cohency, the 95% prediction interval width is 5.1, and we have some outliers right now. And for Shell Rock, the 95% prediction interval width is 5.5. And for New Bed, the 95% prediction interval width is 5.8. We have quite a few outliers for New Bed, and we have found outliers in less than 0.02% of the data, and work is ongoing to remove them. And we think it is because of sensor fouling. And then I'm going to talk about model testing. So we needed to validate the regression models, and we had an independent dataset from Haskin Shellfish Research Laboratory, which we have used. So here is the model testing for Hope Creek. The first graph is what I have shown you before, and the second graph is we have used the same model with Haskin salinity measurements at Hope Creek Station, and the mean square error was 1.7. For Arnold station, the mean square error was 1.8. For Cohency station, the mean square error was 2. For Shell Log, the mean square error was 1.7. For New Bed, the mean square error was 2.6. So here is the preliminary model validation summary. Here is the equation and the name of stations and 95% prediction interval, mean square error, the value of ABC, and distance from Reedy. Here we can see that from Hope Creek to Shell Rock, I am within my goal of predicting the salinity history with the accuracy of plus minus 2 PSU. But for New Bed, the mean square error is 2.6, which is fair because it is the farthest away from Reedy, 39 kilometers to be precise. Now I'm going to talk about salinity estimates for SIGO program. So the salinity estimates for SIGO, our goal was to apply the model to estimate the salinity experienced by oyster before collection. For that, we needed to calculate the maximum number of consecutive hours of salinity below 5 from April 2018 to April 2019. So the method for doing that was to use the regression model and salinity of Reedy to estimate salinity at Army station from April 2018 to April 2019, and then taking the hourly average of Army predictions, and then find out how many consecutive hours of with salinity was below 5, and then compare model result with observation. So here is the model prediction results. I'm going to walk through this graph. So in the x-axis, we have time from April 2018 to April 2019. In y-axis, we have salinity. The first graph is for Hope Creek, then Arnold's, then third is for Cohency, and fourth is for Shell Rock. So the blue lines are representing always Reedy. The orange line is for Hope Creek salinity measurements, and the green lines are for Arnold salinity measurements, and the red line are for Cohency salinity measurements, and the pink line are for Shell Rock salinity measurements. And the black dots are hourly averages, and the salinity, I have put a black line for salinity 5. So then we thought that as we have real life observation from April 2018 to December 2018 to compare the model result with the real life observation. So for Hope Creek 2018, here in the x-axis we have time, in the y-axis we have salinity. The blue lines are representing Reedy salinity, and the orange lines are for Hope Creek salinity, and the black dots are hourly averages. First graph is for model result and second graph is observations. So here we can see that whenever Reedy salinity is very low, the model loses its prediction power, but it's not too bad for Hope Creek. Still, the model is going to give us a somewhat correct answer for how many hours of salinity was below 5. For Arnold, the green lines are Arnold salinity measurements, and for Arnold also same similar type of story as like Hope Creek. Even though the model loses its prediction power when Reedy is very low, Reedy salinity is very low, the model is still going to give us the almost correct answer. But for coherency, there are at least 3 times when salinity was below 5, but the model did not capture it at all. So why is that? To explain that, I have focused on this area. So here we can see that in the model result, the coherency is almost constant, but in observation, there are variations. Why is that? So what is the function? In the simplest form, function is y is equal to mx plus b. So y is here in my model, y is oyster bed salinity and x is Reedy salinity. So whenever Reedy salinity is very low, the mx is going to be close to 0. So we have only b. So that's the constant. So it's almost like a constant, but in real life we have variations, and we have yet to find out why is that. So for Shell Rock, also similar kind of story as coherency. There are at least 2 times when the salinity was below 5 and the model could not predict it. And for new bed, fortunately for us, salinity is very high. So the salinity below 5, there are no occurrences. So now I'm going to talk about our next steps. Our next step is to try to fill in the prediction when Reedy is less than 0.3. We are now going to use temperature and then investigate why the best fitting model is a log function. We are going to find out the physical processes behind this, and then compare our result to previous works and collect data this summer for further model validation. Thanks to Dr. Daphne Munro, Dr. Larry Sanford, and Dr. Matthew Harry, and Iris Bird, and NSF. I'm ready for questions, but if anyone wants to contact me, they can contact me in this email address. Thank you.
There's plenty of time for questions, Kurt, if you want to do questions. Oh, let's go.
Yeah, as a reminder, feel free to put in questions in the chat box. Nothing quite yet.
If we don't have time for questions, we will wait for 1 more minute until we start our next presentation. Oh, I see one.
So Archie, can you talk more about the logarithmic match?
Yes, what?
The logarithmic match?
Yes. So about the logarithm, why the log function works?
Yes. Yes.
So our thinking is that the log function is because of the shape of the Delaware Bay, because it's a funnel shape. We think the log function works, but we are still finding out various reasons behind that.
All right. We have another question. Are all the measurements and predictions at the same level in the water column?
Yes, they are like Reedy is 1.652 meters, and for the Armicos it's the 0.5 to 1 meters depth.
Okay. A couple more questions. Are the field data for 2018 to 2019 public available? If yes, could you provide the source?
It's not publicly available. The Reedy dataset is publicly available, but the Armicov dataset I don't think is a publicly available dataset.
All right. And if you could get any additional data, what would it be? What are you most interested in?
So we are interested in wind, and we have looked into wind and river discharge. We will also look into sea surface height, and we have looked into tide also. And sea surface height will be our next target, and temperature.
Des, do you have a question?
Yes. Can you hear me, Archie?
Yes, I can.
I noticed it seemed that the problem, or part of the problem with prediction, was that the Reedy point salinity could not go lower than 0. So as you pointed out, it was constant, and so therefore it could not predict a salinity below a certain amount for, say, the Cohansey bar there.
Yeah.
Because it can only go as low as 0. So
Yeah,
that keeps Cohansey at a constant level for that period, right?
Yes.
So I guess you've got to, do you have a way to deal with that problem, do you think? Or maybe I missed that.
So we are yet trying to found out that we have tried many things, but we are not still successful. Right now we are trying with temperature.
I see. Thank you.
You're welcome.
Okay, move on to our next speaker.
Okay.
Thank you.
Thank you all.
Okay. Well, I cannot seem to start my video here, but oh, now I can. Thank you, Archie. And our next speaker is Janine Barr from Rutgers University. Her title is Nutrients, Salinity, and Oysters: The Impact of Extreme Precipitation Events on Eastern Oyster Ecosystem Services in the Delaware Bay. Go ahead, Janine. You might need to unmute.
Yeah, I was. Thank you so much.
Okay.
Can everybody see my screen okay?
I can see it.
Great. Thank you. Okay. So hi, everyone. My name is Janine Barr, and I am a second-year master's student at Rutgers University. My research focuses on quantifying the role wild and farmed eastern oysters can play in mitigating nutrient pollution in the Delaware Bay. And I'm really excited to be here today to highlight some preliminary findings from this effort, which was a collaborative effort with all the amazing scientists that are listed here on the bottom right side of your screen. Okay. Reports indicate the Delaware Estuary has high nutrient loadings and high concentrations of nutrients that could yield suboptimal ecological health for local water bodies, including the Delaware Bay. Oyster populations in the Delaware Bay could help mitigate these growing nutrient concerns and supplement existing nutrient management strategies in the region. This is because oysters are suspension feeders, meaning oysters improve water quality by removing particulate matter from the water column when they feed. Now, oysters are resilient critters. They are generally tolerant to changes in salinity, which they see on a daily basis in the Delaware Bay. These daily changes are being shown in the now hopefully moving figure on your screen to the right. This is a NOAA-generated map of predicted surface water salinity in the Delaware Bay over a span of 48 hours from February 18th of this year. Warmer colors represent higher salinity conditions. So what you're seeing is that salinity changes naturally daily from incoming and outgoing tides and are influenced by the influx of fresh water, primarily from the Delaware River. Something else to note about oysters and salinity is that oysters are sensitive to prolonged low salinity conditions, which cause severe mortality events to populations. These low salinity conditions are primarily triggered by storms, which bring large amounts of fresh water into the marine ecosystem. Unfortunately, the frequency and intensity of storm events are expected to increase with climate change. This increase in freshwater input could create more acute low salinity events in the Delaware Bay and alter oysters' ability to provide those water quality benefits. For these reasons, I am interested in understanding the water quality benefits oyster populations provide today and how climate change could impact those water quality benefits moving forward. So the goals of my research are twofold. The first goal is to estimate the baseline level of water quality improvement provided by wild and farmed oysters in the Delaware Bay. And the second goal is to estimate the impact of low salinity conditions on the oyster's ability to provide those water quality benefits. To quantify these benefits, we measure oyster filtration rate. Filtration rate is the biomass of particles that oysters remove from the water over a unit of time. And it's important to note that oyster filtration rate is known to vary in response to a number of local conditions like food quality and quantity, water temperature, oyster size, and salinity. However, site-specific research regarding the impacts of salinity to oyster filtration are lacking. And although we know that filtration is variable, average rates are often used in estimates of ecological filtration. So there really is a need for accurate estimates of filtration that account for all this variation. First, I'll talk about how we estimate the baseline level of water quality improvement oysters provide. So to achieve this baseline understanding, we conduct filtration rate experiments in the field. Our field sites are shown in this map to the right of the southern end of the Delaware Bay. We conduct experiments at one oyster reef, seen here towards the north in the wild box, that's the location of the wild reef, and one oyster farm seen here to the south in the farm orange box. And it's also depicted in the picture to the left, which is an intertidal rack and bag oyster aquaculture system. The wild reef is subtidal, so I unfortunately don't have a photo of that to show you guys. We conduct the filtration experiments in situ rather than in the lab, so we can expose oysters to conditions as close to their natural environment as possible, which will generate more accurate filtration results. The filtration experiments are performed up to 6 times through a calendar year at each site to capture those seasonal variations in oyster filtration. To conduct these in situ experiments, we use a flow-through experimental chamber developed by Yves Galimani to collect filtration information from up to 18 oysters at a time. The chamber is shown here on the right side of your screen. Local water is pumped into the chamber through this orange hose and then flows into these smaller chambers on either side of the main chamber at a predetermined flow rate. Each small chamber holds 1 oyster, so I call the tiny chambers studio apartments. And just to highlight how water flows through the chambers, you can see the schematic on the left, which shows how water flows from the main chamber into a studio apartment by following those black arrows. Water samples and oyster waste, or biodeposits, are collected over the duration of the experiment. The water samples provide information regarding what is entering each oyster, while the oyster biodeposits, which are called feces and pseudofeces, provide information regarding what has been excreted by each oyster back into the environment. So what you can see in this slide here is that feces are those more ribbon, dark-like structures close to the green feces label, and pseudofeces are more cloud-like, pillowy biodeposits, if you will, shown here, and also in more close-up images to the right. By understanding what is entering and exiting each oyster, we can calculate what each oyster absorbs and thus determine filtration rate. Once enough biodeposits have been collected, the experiment is terminated and the samples are returned to the lab for processing. This includes filtering the water samples and biodeposits through glass microfiber filters, which are then burned and ashed to determine organic content of each sample. I also wanted to highlight that for each experiment, we do measure the amount of time it takes the oysters to digest food, which is what you see happening here. We do this to ensure we get real results from the chamber. And now that we know how the experiments generally worked, let's look at some preliminary results. So to orient us, this figure shows the results of 2 experiments done on the wild oysters in 2020. The x-axis is the month the experiment was conducted, and the y-axis is the average of all the individual oyster filtration rates. We have also standardized each filtration rate to 1 gram of the oyster's dry tissue weight to account for the impacts of oyster size on filtration. Below this main figure, we have pie charts to show the proportion of oysters that were closed during the experiment and thus were unable to collect filtration data on. Here, we see that the average filtration rate of wild oysters in August was higher than the average filtration rate of oysters in October. Now, this shows us a relationship we'd expect to see. Oysters have higher filtration rates when the water temperatures are higher, but that's up to an optimum. So if the water temperatures are too hot, they'll actually get stressed out and they'll filter less. But this general seasonal trend is something that we would expect to see. And here are our results from the farm experiments. Again, the time of the experiment is on the x-axis and the y-axis is the average filtration rate of the oysters. The proportion of closed oysters are seen in that pie chart below. Here we see that the average filtration rate of oysters measured in July and October were fairly similar. Of note is the large error bar on the July farm data, and that this warmer July month does not show a higher average filtration rate than the October experiment, which was the expected trend that we saw in the last slide with the wild oysters. We attribute this wonky July data to the fact that this was our very first experiment that we conducted and that we were still in the learning mode with the techniques, and some human error may have been the source of this variability. Here are the farm and wild experiments together, so you can see how the 2 kinds of oysters compare. I'm interested generally to see how similar the average farmed in orange and wild in blue oysters are, or how their filtration rates will be similar or different as we compare all the different results that we will collect in 2021. And also, just to highlight the variability of each oyster's filtration rate, I'm going to use this schematic of the chamber and its studio apartments to show you. So what I'm going to do is overlay each oyster's filtration rate for the August wild oyster experiment to the studio apartment they were placed in for that experiment. The y-axis on this graph and the one that just popped up is the oyster's individual filtration rate per gram of dry tissue weight. So this just shows the variability in each oyster that does contribute to the error bars that we've seen in the previous slides. Okay, so now that we've learned a bit about the baseline level of oyster filtration, let's move on to the second research goal of estimating the impact of low salinity conditions on oyster filtration. To do this, we exposed the oysters from the normal salinity experiments to 3 days of low salinity and repeated the in situ chamber experiments. So we chose 3 days because that was about how long it took the freshwater signal from Hurricane Sandy to leave our area. So we were trying to mimic storm-like conditions for these oysters in these tests. So the general methods can be explained by this figure. Here you will see all 4 normal salinity experiments grouped by season. So the left side has the warmer months and the right side has the cooler months. The oysters in colored circles represent the average salinities for each group of oysters, each set of 18 oysters, and what they were exposed to during their first filtration experiment. So for example, the farmed oysters that were examined in October of 2020 were tested at a normal salinity of about 24.5 parts per thousand. And just to show you all, this schematic does go all the way down to 11 parts per thousand. After each normal salinity experiment, the 18 oysters that were in the experimental chamber were secured in mesh bags like this one and deployed off docks at low salinity sites for 3 days. At the end of those 3 days, the 18 oysters were placed in the chamber again for a second experiment. These boxes that are appearing now represent the average salinities observed during each of the 4 low salinity experiments. So for the farmed oysters, again analyzed in October 2020, their filtration rates were measured at both 24.5 parts per thousand and then 3 days later at 15.9 parts per thousand. By conducting these paired normal and low salinity filtration experiments, we were able to generate some interesting comparisons. And before we move on, I just want to note that there was a really small difference in salinity for these wild oysters that were tested in August. This slide shows the results of these low salinity experiments. This box plot shows the difference in oyster filtration rate between their normal and low salinity conditions. The x-axis has the 4 paired salinity experiments, while the y-axis plots the difference in each oyster's filtration rate. Now, what's important to know about this figure is that if the colored boxes representing the different kinds of oysters are mostly above this red 0 line, we know that the oysters filtered more at their normal salinity than at a low salinity. So what we see here is that 3 of the 4 sets of experiments show higher oyster filtration rates under normal salinity conditions than at a low salinity. And these asterisks in red over 2 of the experiments indicate a significant difference in filtration rates via a paired t-test. So here again, unfortunately, we're seeing the results of a wonky July field day because these oysters do not show the same trend. In fact, this indicates that the oysters actually filtered more in low salinity than high salinity. But again, this wonky result can be attributed to the human error of us still being in the learning phase of experimental protocols. But this was really exciting because it shows that filtration can significantly decrease during low salinity conditions. And remember how I said that the August wild experiment shown here did have a small difference between the normal and low salinity conditions. I just want to mention that I am processing and analyzing other environmental data to tease out if salinity really was the factor that caused the change in filtration rate. So things like temperature DO are things that I'm looking at.
2 minutes, Janine, 2 minutes.
Thanks, Sarah. So just to wrap up, here a summary of the preliminary findings from this work. Oysters exhibit a decrease in filtration rate as air temperatures cool, which is something that we saw in the baseline oyster data. We've also seen that short-term exposure to low salinity conditions can significantly impact an oyster's filtration rate in those salinity experiments. And just overall, we found that each oyster reacts differently to its environmental conditions. So it's important to compare those same 18 oysters at their normal and low salinity, definitely. And in terms of future work, we are planning to conduct experiments through the summer of 2021. That is just to get that full year of seasonal variability. Also, as I mentioned, I'll be analyzing the influence of other environmental variables on oyster filtration. And I'm also planning to calculate nitrogen removal rates for these oysters. That is data that was collected at the same time as these chamber experiments, but they aren't analyzed here. So to fully wrap up, I just want to thank the wonderful people, the collaborators and the funders who helped not only make this work possible, but fun and exciting. So thank you to everybody who helped make this happen.
Fortunately, no time for questions. Sorry, but maybe at the end. Des, next speaker.
Ooh. Very good. Let me see here. Can you hear me? No?
Yep, yes, yes.
Okay,
You're
good. Thank you, Janine. Okay, our next speaker is Manuel from the Rutgers Haskin Shellfish Research Lab. And the title of her talk is Habitat Selection Has Implications for Post-Settlement Growth of Oysters in Different Situations. Go ahead, Emily.
All right, hello.
Make sure you unmute.
Yes.
It looks good.
Okay. Sorry. Good. All right. So, hello everyone. As Des so kindly introduced me, today I'm going to be telling you about how initial settlement habitat has implications for growth of juvenile oysters in different salinities. Throughout this talk, I'm going to be referring to juvenile oysters as spat, and you can see a spat that's set on an oyster shell in the picture on the left. And I always just like to clarify what a spat is, because when I first showed a picture like this to my friend, he was like, oh, they're a lot bigger than I thought. But that's because he was looking at the whole shell, when in fact the spat is really just that one that I have circled. So Delaware Bay is an estuary in the northeastern United States. It is tidally controlled between the Delaware River and Atlantic Ocean and home to the eastern oyster. For the purposes of this study, I have grouped the fishery management regions into salinity zones. And during this presentation, associate a dark blue color with high salinity and a light blue color with low salinity. These zones have distinct characteristics: slow growth and low mortality in the low salinity zone, and fast growth and high mortality due to disease and predation in the high salinity zone. Oysters can't move post-set like a few other bivalves can, but the environment can change around them. So for fisheries management, oysters are moved from up bay in the low salinity zone to down bay in the higher salinity zone because it encourages oysters to grow faster and therefore they can be harvested sooner. Another way is through climate change, which increases the frequency of storm events, which decreases salinity. And the question I'm trying to answer is, how does the initial salinity habitat affect growth after an environmental salinity change through one of these two methods that I just described? So keeping that question in mind, we wanted to answer to determine how initial salinity conditions carry over into additional growth in different environments. We had a few hypotheses to explore with these data. First, we expected the least amount of growth from spat in the lowest salinity treatment, which is exactly what we observe in the wild. Those up bay and the low salinity grow less than those down bay, or grow slower than down bay. Our next hypothesis was that there would be an interaction between salinity zone and salinity treatment, with spat growing the most in the salinity treatment that is closest to the salinity zone from which they came. This is demonstrated in the graphic on the right. You have higher growth from the oyster that does not move to the different colored rectangle. So you can see here, I expected higher growth in when they didn't move, but when they were transferred, I expected lower growth. And when spat first set, they grow flat against their substrate, which you can see in these top two pictures. As they grow, they pop up into the three-dimensional oyster shape. So in this picture, you can see kind of their transition where you see the edge just starting to creep up. And then in this picture here, you can see that it's fully popped up. You know, it's got the cupped shell and everything now. So we lastly hypothesized that flat spat would have increased growth because they were only growing in 2 dimensions rather than growing in this third, like height dimension as well. So I designed 2 types of experiments to answer these hypotheses. My first type of experiment I will refer to as the wild experiment. As the name suggests, spat were collected from the wild and placed into experimental laboratory salinities. In 2019, spat were collected from 3 different beds covering the high and moderate salinity zones. They were crossed into 3 salinity treatments that corresponded to salinities experienced in each salinity zone. In 2020, spat were collected from 7 different beds covering high, moderate, and low salinity zones, so from the whole bay. And they were crossed into 1 treatment that corresponds to the high salinity zone, 1 that corresponds to the moderate salinity zone, and then 2 that were corresponded to the low salinity zone for a total of 4 treatments. For both of these experiments, spat were measured in length and width for 6 weeks. So first, I'm going to orient you to my graph. I have daily specific growth on the y-axis and the different salinity treatments on the x. My plot is faceted by the different years. So we got 2019 on the left, 2020 on the right. And the colors correspond to the different salinity zones from which they came from. And using pairwise comparisons with my high salinity treatment, which in this case was 22 parts per thousand from the 2020 experiment, I compared the other salinity treatments and determined that there was not a significant difference in growth based on salinity treatment. Also using a pairwise comparison with my highest salinity zone, which is the dark blue color, there was a significant difference in growth from spat in the lowest salinity zone, but not with the moderate salinity zone. So that's primarily looking at this 2020 data. There were no significant interactions between salinity treatment and salinity zone, but the shape of the oyster, whether it was popped or flat, did have a significant influence on growth, with the flat spat indeed having higher growth than the pop spat. So even though the spat were collected on the exact same day, both on October 1st in 2019 and 2020, there were likely some environmental differences between the years that were not controlled for, which could have influenced how the spat grew in the experiments and minimize the effects of the salinity treatments by the time I got to them. Spat from the lowest salinity zone had highest growth between the salinity treatments, most likely because they were moved into higher salinity treatments, which removed osmotic stress and they were just able to take off. There was not a significant interactive effect, and spat did not have highest growth in the salinity treatment analogous to their salinity zone. This could be because they were used to regular salinity fluctuations with the tide, and as long as they were above the minimum threshold of about 5 parts per thousand, they were able to compensate. Flat spat showed higher growth than pop spat because length and width do not fully capture growth in the popped spat. But since all of my spat were set on substrate, you couldn't measure it accurately. So for my next type of experiment, I'm going to refer to it as my hatchery spat experiment, which actually occurred between the 2 years of wild experiments. And I performed the hatchery spat experiment to isolate the effect of salinity from other environmental variables associated with field collected spat. 2 oyster groups were spawned and raised in 22 parts per thousand. One group was a hatchery-bred, disease-resistant oyster, which I will refer to as the selected group. The other group was a cross between the selected line and wild oysters, and I will refer to that group as the hybrid. So once the larvae reached competency to set and metamorphose into spat, I placed them into 4 salinity treatments and let them set and grow in the initial salinity condition for 3 weeks. And after 3 weeks, they were photographed and then photographed to be measured and then crossed into new salinity treatments. And they grew an additional 3 weeks before they were photographed to be measured again. These spat were much smaller than the wild spat. I don't even know if you can really see on your screen, but just for comparison, these were pretty different. So this figure shows the area of the spat on the y-axis and the type of spat on the x. It is colored by initial salinity they were placed in, which is analogous to the salinity zone from the wild spat experiment. So yeah, during this time period, growth in the 22 and 16 parts per thousand, so these 2 darker colors were pretty similar. And then across both types of spat, and then in the 10 and followed by the 10 and 6 overall. And the takeaway from this is that the spat types grew differently, and then there also is a trend of decreased growth with decreasing salinity. So similarly to the first graph I showed, this figure has daily specific growth on the y-axis and salinity treatment on the x. It is faceted by spat type, and the colors again represent initial salinity, which think about it as similar to salinity zone. So in the hybrid group, and the final salinity treatments show similar growth in the 22 and 16 treatments, which decreased for the 10 and 6 parts per thousand treatments. In the selected group, there was a decrease in growth as salinity decreased, and in both groups across all treatments, highest growth occurred from spat, which were in initial salinity 10. The initial salinity treatment significantly influenced growth, but final salinity did not, and there were significant interactions that occurred with initial salinity 10 or with final salinity 6. There was also a significant difference in growth between the 2 spat types. So in the picture on the left, you can see an example of a spat that was in initial salinity 10, then transferred into 22 parts per thousand. It's about 3 or 4 times larger. And the significant difference in growth between spat types is likely because the selected spat were set a week later, but placed in the same tanks as the hybrid group during the initial salinity period, so they were less likely to compete for food as efficiently. This initial size difference allowed for faster growth in the second half of the experiment. Also with the selected type, no spat that were in the initial 6 parts per thousand treatment lived until the end of the experiment, even those spat that went from 6 into 22, for example. However, those that were in higher initial salinities, but then transferred into lower salinities were able to survive. It is unclear why the 2 larval type perform differently, and further work needs to be done to answer why salinity tolerance between the selected and hybrid groups differed, but that was not the takeaway from this experiment. So thinking about the results from the experiment, it is clear that initial habitat conditions have a large effect on subsequent growth, and this carryover effect might be responsible for masking the effect of growth in a changing salinity environment. In a broader context, when the fishery makes decisions on where to transplant oysters for faster growth, these results suggest oysters transferred from low salinity might grow faster than oysters that were always in a high salinity environment, at least for 3 weeks, which is the duration of my experiment. So I would like to thank the technicians who did a lot of the daily maintenance for these experiments, as well as the NJDEP for use of their boat to take me out to collect their oysters. This research is supported by NSF under lead PI Matt Hare. And lastly, I would like to thank my SPAT for their invaluable participation. Thank you all for coming to my talk today, and feel free to reach out to me through my email, which is on the bottom. And with that, I would be happy to take any questions that you may have.
Thanks, Emily. We do have time for some questions, if there are any. Kurt.
Nothing so far. So certainly post in the Q&A box or save it for the end.
Okay.
I see one, Kurt.
Oh, here we go. So Emily, can you please explain again the difference between the hybrid and selected groups?
Yeah, so the selected group is the Rutgers NEH disease-resistant oyster. It's pretty popular among aquaculturists because they obviously don't want their product to be killed off by disease. So this is, so that's that line. It's selectively bred for that disease tolerance. And then as you likely know, with you want a little bit more of introduce some new genetics. So that's what the wild oyster hybrid was brought in to do, was just so that way you don't end up with some negative like inbreeding effects. So those are the 2 groups.
Okay. How does it inform leasing areas?
Yeah, that's a great question about kind of bringing it more to aquaculture. A lot of aquaculture happens in a higher salinity environment because, as I mentioned, it does generally to higher growth. However, with aquaculture, there's obviously a lot more input. You know, the farmers are going out there and tumbling their bags and like cleaning them off and all that kind of stuff. So I think this might not have as much to do with the leasing grounds, but maybe thinking about where they buy their seed from and just how that might help influence their growth later on. Thinking more about where the seed came from and those initial carryover effects might be beneficial for their specific lease. So thinking about both of them as a group rather than just the site.
Okay, I think time for maybe one more question, Sarah.
One more, yep.
Okay, and Dave, we'll get to your question at the end of the session. So Emily, how has these data compared to the other geographic areas on the East Coast?
I have not compared it to any other areas on the East Coast. That could be another part of the project, is getting a broader range of not just the 2 spat groups that I used, but maybe pulling from different hatcheries and repeating this experiment. So I will get back to you on how it might have a broader context with that.
Thanks, Emily. And we have our final speaker. It's a lightning talk. Des?
Yeah. Okay. Hold on just a second. Thank you, Emily. And our final speaker is Heidi. And her topic or title is Seasonal Oyster Microbiome Dynamics at Low and Moderate Salinity Sites in the Delaware Bay. Go ahead, Heidi.
Thank you, Desmond. To ensure that this lightning talk has lightning speed, I will be sharing a pre-recorded presentation. I will hop off here after I share my screen.
Hello, everyone. My name is Heidi Yeh. I am a PhD student at Rutgers University, where I completed the following work with my advisor, Dr. David Bushek. Today, I will be presenting some preliminary data on oyster microbiome dynamics driven by salinity and seasonality in the Delaware Bay. The microbiome, which is the community of bacteria that live on and within an organism, is influenced by many factors that are external and internal to its host. In the case of the oyster, filter feeding activity makes for a particularly strong connection between oysters and the waters they are submerged in. As a result, they are influenced by salinity, temperature, ambient microbiota, pollution, and more. There is an interplay between these external forces in the internal dynamics of the oyster. For example, oyster immunity is compromised by higher salinities. The preliminary data I will present today explores the impact of salinity as well as seasonality on the oyster microbiome. Back before the pandemic, from July to October of 2019, oysters were collected on a monthly basis from 2 sites in Delaware Bay that differ markedly in their salinity levels. The sampling coincided with the peak of dermo disease prevalence in the bay, and future data analysis will look at the potential interplay of microbiome dynamics with the disease. Bacteria from the collected oysters were plated and incubated overnight to enumerate bacterial abundance based on colony forming units. Bacterial abundance is represented by colony forming units here on the y-axis. All marine heterotrophic bacteria are shown in this plot. With the exception of August, bacterial abundances were very similar between sites and declined with time as water temperatures cooled. There was much more variability in the number of bacteria from the Vibrio genus, shown in the right panel, which I selected for with TCPS medium, but numbers were generally higher at low salinity sites. This is consistent with previous studies which have concluded that Vibrio pathogens of concern for human consumers are reduced in oysters held in saliniters. With the collected oysters, digestive glands were dissected out. This is one of the tissues with the highest bacterial abundances in the oyster and also has a more stable bacterial community. DNA was extracted and the 16S rRNA V4 hypervariable region was sequenced with the Illumina MySeq to identify the bacteria found therein. I performed the bioinformatics with the mother pipeline and various R packages. Alpha diversity refers to the mean species diversity, which for both salinity sites increased over the course of the sampling time period. You can see the inverse Simpson on the y-axis, which increase over the different sampling months. My guess is that as temperature decreases, a more rich bacterial community is able to flourish as fast-growing bacteria that dominate during the summer months slowed down. This plot shows the taxonomic identity of the sequenced bacteria broken up by collection month and the salinity of the site they were collected at. Each sampling had 6 oysters used as biological replicates, so this plot shows the average relative abundance of sequence reads belonging to each taxonomic group. On the y-axis, we can see the relative abundance of the sample that each taxonomic group represents, and all samples show a dominance by roughly 2 to 4 taxonomic groups, and a good amount of agreement between samples collected in the same month, regardless of the salinity site they were collected from. The waning influence of the Tenera cuties as time progresses, that's here in the pink color, gives some support to my hypothesis that alpha diversity increases as bacteria that dominate in the warmer conditions of the summer months decline with declining water temperatures. Looking at the beta diversity of the samples, which is the similarity of the samples to each other when the composition and the weighted abundance of taxa are taken into consideration, such as with the theta YC dissimilarity measure. In this plot, the collection month is indicated by color and the salinity is indicated by shape. Samples are most tightly clustered in the month of July and become progressively looser in their associations over the season. Salinity does not seem to have any organizing influence within this plot. In conclusion, season had a notable influence on the bacterial composition and alpha diversity of oysters. Past work shows that there are denitrifying bacteria as part of the oyster microbiome, indicating a potential role in the nitrogen cycle, which I plan to explore with qPCR analysis of genes involved in denitrification. Additionally, Vibrio abundance has a weak inverse relationship with salinity. Expanding the scope of this testing beyond this study period of just a few months to understand more about the relationship between salinity and Vibrios of concern to human health could inform regulations around reef restoration projects. Here are my references, the first to a paper that supports the connection observed between salinity and pathogenic Vibrio abundance, and a review paper referenced in the intro that summarized internal and external influences on the oyster microbiome. And I would like to acknowledge all of the parties involved in the collection, sequencing, and funding of this project. Thank you all for your time and attention. Hello.
Okay, that's it for me.
Okay, we'll just open it up to questions for all our panelists and speakers. If anyone has any questions, hand it to Kurt.
All right, so first we have one for Heidi. Could you briefly explain what the mother pipeline does?
Yeah, the mother pipeline takes the raw sequence reads from the Illumina MySeq machine, and then we can create our own custom scripts to be able to target the region and maybe use a database that is specific to what we're wanting to look at to just make the analysis more efficient. Yeah, so basically it's just to identify the bacteria that are present and then to prepare the data for use in our packages.
Okay, and one more question. Can you explain in a little more detail what you mean by alpha diversity?
Yeah, alpha diversity is just looking at each individual sample, whereas beta diversity is looking at them all together as a full data set. So with the alpha diversity, there are different they give weight to different things. So some of them might just look at how many species are present. Another might look at how many species are present and then how much of the population is represented by each. So like in the summer months, we saw that perhaps there were a lot of things present, but one tag was really dominating things. So therefore it had a low species, a low alpha diversity.
You're muted, Kurt.
Sorry. Yes, we had a question for Emily. Did you see spat morphology differences occurring at different sizes at high and low salinity?
No, not when I got them in from the fields. There was a mixture. It wasn't like only those that came from the low salinity zone were all flat. There was I can show a figure if I can share my screen again, but I don't want to take up a bunch of time.
Feel free to share the screen. We can ask another question in the meantime. Janine, is there a difference in nutrients along the salinity gradient and how that might influence their filtration?
Hi. So the question was if there's anything inherent about the two locations that would make their filtration rates different. Oh, like water quality data.
Right, nutrients along the salinity gradient.
Cool. Thank you so much for your question. So I have not found, I haven't analyzed the data that we've collected specifically about nutrients in the two different areas. Something that we do to analyze that is take nitrogen samples of the actual water that the oysters are living in. So once I have that data, I'd be happy to share it. But something else we do at each site is we take raw water samples and we run it through something called a flow cam, which is basically a high-tech microscope-like device that takes images of water samples very quickly and can capture very small particles that are in that water. And so we have information available to us about like what the quality of the food is that they're eating. Presumably, if there's a lot of stuff that they can't eat, their filtration rate will be higher because they have to sift through a bunch of non-edible things to get their nutrients. But that is something that I will look at, and I appreciate the question.
You're muted again, Kurt.
Oh, sorry about that. I think it's Emily. Go ahead and share what you're going to say about your screen.
Yeah, so I think Dave's question was just if the salinity zone had influenced their shape, if I'm remembering it correctly, and no, and this proportion of weeks popped. So, so if they stayed flat the whole time, you know, that's these guys over here, and if they were popped the whole time, that's these guys over here, and you can see the general negative trend with as they the amount of time they were like popped or not. Yeah, so I I think I answered Dave's question with this graph.
All right, anyone else? We're almost out of time. Oh, Janine, could the low salinity treatment that elicited higher filtration rates have had different food quantity, e.g. more turbid?
So thank you for that question. I think for that low salinity, you're referencing the oysters that did have a really wonky normal salinity data collection day. And based on just looking at the water between those oysters' normal salinity site and the low salinity site, the normal salinity site was much more turbid. It's in like this coastal mudflat area of the Delaware Bay. So it's like pea soup. You can barely see through it. Whereas the low salinity site was actually much more clear water. So I like where you're headed with that question, but unfortunately, I don't think that that was the cause for the weird filtration rate results for that day. I can talk to you more about it later, but there are some pretty clear indications in another set of analysis I did on that first normal salinity day that show that it really was human error that caused the problem.
All right, I think we're good.
Yep. Thank you, everyone, for coming. Thank you, Des, for moderating. Just throw two quick things in the chat real quick. Please check out our sponsorship. Our sponsorship profiles. Couldn't do this whole summit without them. And then our poster session is coming up in just half an hour. So if you can check out our posters and pre-recorded presentations before then, that would be good to help support conversation. But thank you to our speakers, and I'll see you in another session.
Thank you. Thank you, everyone.
Thank you.